Welcome to the second installment of the Newton webinar series. Did this a couple of weeks ago and had a chance to talk to Benoit Bruneau about how genes sculpt the heart. And today we're going to talk to Steve Finkbeiner about the Thinking Microscope and how AI is being brought to bear on his work and on biomedical research more broadly. If you weren't on the first webinar, just to give you a sense of what we're trying to do with this, first of all, it was meant to be a respite from all the COVID-19 content that we're all consuming, and just a reminder of the breadth and depth of our research at Gladstone and across the scientific community. Also meant to give you some insight into the scientific mind and how they think, how the labs operate, what the processes and programs and all of that are behind the discoveries that we wind up reading about. There's a lot of hard work that goes into a breakthrough. And so we just want to give you some insight, a window into that. So today, one thing I would say at the beginning is, as we're talking, if questions occur to you, there is a Q&A function at the bottom of your screen, and you can post questions, and we'll block time at the end to get to those, as many of those as we can. So Benoit last week. Today, Steve Finkbeiner, really one of Gladstone's most original and creative thinkers, as you'll see. I was thinking about this morning, really a kindred spirit with Newton. If you remember Newton's work on his reflecting telescope, he really spent most of his career trying to get that thing right, and grinding lenses and developing prototypes and throwing them out. He died in 1727 or thereabouts, and right at the end of his life, somebody else essentially perfected the optics in that microscope or the telescope, so he got to see it fully realized. This is part of Steve's work, is building the tools that he needs to do his work, as you'll hear. So I have a sense that if Steve lived in the 18th century, he definitely would have been hanging out with Newton. So Steve, welcome. Where are you? I see your background. I think yesterday you were in the Hong Kong gardens or something. Where are you today? Steve, can you hear me?
Oh, there you go. Sorry.
Yeah, there you are.
Yeah. So that's actually the place where bungee jumping started in New Zealand. I jumped off that bridge a year ago, and you get a t-shirt for doing it, but my wife grabbed the t-shirt because she thought she deserved it more than I did. So
I was thinking of you this morning because sheltering in place must be especially challenging for you because you're so itinerant. You flew, what did you say, 387,000 miles last year. I think that number has gone down this year. How are you weathering all this? And how are you doing science by Zoom?
Yeah, well, actually, 2 hours before this, I gave a talk to 500 people around the globe through Zoom. So it's, it isn't as fun, I think, as face-to-face, but it's really clear that the scientific community is eager to get together in any kind of circumstance, and there are some great questions and a lot of stimulating discussions. So I think we're trying to muddle through this as best we can.
Do you think it's going to change the way science operates? I mean, are we developing different muscle memory? Are there silver linings maybe to this change, or do you think it's just a muddle through and then get back to normal as soon as possible?
Yeah, it's a great question. I think a lot of us scientists are spending maybe the majority of our days on Zoom or some other video conferencing platform. And I would say that we had been using those tools before this shelter in place, but I think we've gotten a lot better at using them. And I think because we spend so much time on them, we are beginning to approach them and kind of think creatively about how they might be used in ways that we weren't able to do things before, as opposed to kind of the tool of last resort. And so I was really struck this morning. I think even though I flew about 400,000 miles last year, that's still only a limited number of places in the world.
Yeah.
And so this ability to do Zoom with literally an unlimited audience number has a lot of potential in terms of both reaching out to the global community. And I also think, I was just thinking about this morning, there are, segments of our society, underrepresented groups that often don't get visited by scientists. And so I think this could create a lot of opportunities to democratize science, I think, in a way that we haven't had before. And so I think I am kind of excited that rather than just going back to the way things were before, I think we all now see a new tool in our toolbox. And so I think we're going to think creatively about how this could be.
That's super interesting. I hadn't thought of the equity issues. I don't only really thought about the negative of, people it's harder for some folks to raise their hand on this format, and there, are some patterns developing that are a little discouraging, but we're reaching more people.
Yeah.
So that's really interesting. I know we're going to talk about your microscope and about your work in general, but wanted to spend just a little bit of time figuring out how you got here. I know you're a Midwestern kid and sort of grew up bashing your head against people playing football. How did you go? So how did you get to Wheaton? And so tell us the story a little bit. How did you become the scientist you are?
Yeah, well, like Robert, like you said, I grew up in the Midwest, a suburb outside of Chicago, but my mom came from farmland in North Dakota. So I used to spend the summer on farms in North Dakota helping out for harvest and things like that. And I think there's a resourcefulness in the Midwest sometimes. A lot of times, especially on the farm, if you can't solve a problem, your nearest help is about a half an hour away. And so it definitely drives a lot of creativity.
If the tractor breaks, you can't wait.
Yeah, pretty much. You're highly motivated to figure out a solution. I'll just say that.
Yeah.
It's hot and it's humid and helps a long way away. So I think that, and I think being around all those things made me unafraid of taking things apart, putting things back together, trying to understand how things work. So I was adopted. My parents, neither one went to college, and it turned out I was kind of smart. So they figured, well, smart kids become doctors. And so that was kind of the direction they encouraged me to go when I was young. And
well, I think of Wheaton as a liberal arts college.
Yeah, so Wheaton is a liberal, it's a small liberal arts Christian college in the Midwest. And my family, we come from a strong faith-based kind of community. And so that's kind of how I ended up at Wheaton. But yeah, really discovered that just fascinated by science and interested in medicine. And so went to Yale University to do my medical school. And they have a special program there called the MD PhD, or Medical Scientist Training Program. And if you can believe it, they will pay you a salary as well as your medical school expenses if you do both degrees at once. And so
it's only an option. Isn't it an insane proposition to do both at once?
So it is certainly challenging. What I will say, though, at Yale, which was so wonderful, was that they had a very creative approach that allowed you to intermix your medical school and your graduate school work in a really effective way that made it possible, at least for some of us, to do those degrees faster than you could elsewhere. So I in fact finished both degrees in 5 years, which supposedly is a land speed record still at Yale. But got a little lucky. Yeah, so, and I just
have a sense that, I mean, at some point, did you have to pick a path and say, I'm going to be a doctor, or I'm going to be a researcher, or did you know going in?
No, I think, I think going in, I was really thinking of purely doing medicine, but I think the experience of doing research in a lab, really neuroscience at the highest level, was just so exhilarating to me. I realized I'd found my true love. And so I knew that somehow or another, I had to figure out how to make sure that remained part of what I did for the rest of my life.
Hmm.
But you're right, a lot of folks who do these dual degree programs have a lot of options, and they can do anything from pure medicine to pure research or something in between. And so a big part of the developmental process for them is trying to understand themselves and kind of what their passions are and how they want to make a difference with their lives. And so where they basically come out that other end.
The decision to focus on the brain, was it just because that was the hardest thing you could do, or how did
Yeah,
it that you gravitated to the brain?
I'm embarrassed to say, I think at Yale they had a mechanism. When I first entered, I was just a medical student, but they had a mechanism to get medical students into research. And even as a medical student, you had to do a little mini thesis, not a PhD thesis, but a smaller thesis. So they had an infrastructure already in place to help students do things like that. And so I had a list of faculty and their interests, and I'd say the areas I was drawn to were cancer and neuroscience. And I just talked to a bunch of people, and the neuroscientist that I ended up working with, I'm embarrassed to say, he turned out to be a really famous guy, National Academy member, almost, essentially deserved the Nobel Prize.
Mm.
But I was really drawn by the fact that he had such imagination, and he was just so charismatic. And I really just enjoyed kind of interacting with him. And so I ended up in his lab and then just totally fell in love with neuroscience in the lab.
Tell me more about that. I'm really interested in the role of imagination in a lab. I think that most of us, outside of the scientific community, can't really appreciate how a lab operates, how decisions are made, how you decide which paths to choose. Tell me something about your lab, and then as I say, the role that imagination plays in your research.
Yeah, I appreciate the question. I think a lot of folks don't appreciate just how important creativity is when it comes to science. I think there certainly are scientific tasks, for example, developing a vaccine to COVID-19, where the approaches are very well laid out and where it is more a focus on execution,
Right.
and maybe not so much on imagination or creativity. But that's, that most of science really would benefit from creativity. And I think even the focus of my talk today, this Thinking Microscope, the seed for it really goes back to literally the first day in my new lab at Gladstone. And I was thinking ahead to a scientific career, and I was thinking about the tools that currently were available to scientists, and really thinking about the questions that I most wanted to answer and how I really couldn't answer them with the tools that were available at the time. And so I think what that encouraged me to do was to forget for a minute what was feasible and possible, and to think instead about what it was I really wanted to do. And then once I decided what that was, essentially work backwards to figure out what would I need to invent or create to make that possible. And so I think for scientists, that is part of the process.
I apologize for the sound of the cat in the background. This is life on Zoom, but we have an ornery cat all of a sudden for some reason. My wife is working on it. I imagine as you mentor the folks in your lab, and as you think about helping them figure out what path to pick, I mean, that seems like a really great starting point to go upstream of it all and just think about what could you imagine might be possible and operate without barriers. I never got that advice when I was a graduate student. It really was pick a project that you think you could make some progress on and do that as quickly as possible. Right? Was a sort of transactional quality. So.
Yeah, and I know that we have to spend attention to issues like time to degree and getting students graduated in a timely fashion. But honestly, for me, the best science is when someone really is passionate about a problem, because it just ends up fueling their creativity, and the science they do will be better, will be more of a breakthrough. And also science just inherently has a lot of days where things don't work out, and having something you're really passionate about will help you get through the weeds until you get that important discovery.
Just one more question about your lab, and then I want to get to your science. As you're recruiting postdocs and people into the lab, are you are you recruiting for imagination? Are you recruiting best athlete? Or are you recruiting for positional need? Because I mean, a lab has a lot of work that needs to get done. How do you approach that?
Yeah, that's a great question. I mean, I think that I'd say that passion, trying to somehow figure out passion and collaborativeness would be the two most important things that we look for in folks. But it does depend a little bit on kind of their level of training and expertise and what they're looking for. For folks who seem to be on a career track, training to start labs of their own, that's what we're really looking for. I don't recruit for positions at that level. I'm really looking for someone who just loves science and has a question that they're really passionate about, sees the opportunities to pursue it in our environment. And, and so we have the opportunity to kind of resource them and enable them to really fly. We do have, you're right, there are some needs, we have to do some blocking and tackling. And so when it comes especially around to engineering and some other dimensions, we do need specific skill sets, coding and things like that. But we do tend to emphasize passion and collaborativeness.
The science, tell me about the portfolio of diseases you're working on and where you think we are and what you think the sort of the central problems or challenges are there.
Yeah, so I'm a neuroscientist, and so I'm very interested in brain diseases. Broadly speaking, we kind of work in two large areas of brain diseases. One, the largest, is the group of neurodegenerative diseases, and those include Huntington's disease, Alzheimer's disease, Parkinson's disease, ALS, frontotemporal dementia, and some less common disorders. But we also do some work on psychiatric diseases as well. Autism in particular, we're part of a large consortium. And because it's the brain, and there are a defined set of cell types, it turns out that some of the mechanisms of those diseases, even though clinically they're very different, do overlap. So some of the tools that we develop to study them can be really helpful. And I personally find it very helpful to actually study a number of these diseases because we do see common threads that emerge by studying them as a group. And I think that's very helpful because it's been so hard to develop therapies for these disorders that finding some common threads, I think, give us a way to focus, especially from the standpoint of developing therapies. Now, we are a neuroscience lab, so that's our primary focus, but some of the technologies we've developed, like the focus of today's talk, AI and robotics, turns out to be applicable outside of neuroscience too. And so we have, in addition to our academic focus, a large number of relationships with drug companies to help them make treatments for patients. And a number of those companies from outside neuroscience have brought us into their areas of disease because the technologies we've developed have so much promise. So areas like cancer and gut disease and some other areas. I think you're muted, Robert.
I am. I got a notice saying I couldn't unmute myself. Am I now unmuted? Yeah.
You are.
I do want to talk, if we have time, about the partnerships that you have with folks beyond your lab, but just on neuroscience in general, or neurological disease research in general, how would you represent where we are? Are we accelerating? Are we just making steady progress? Where are we in space and time in terms of solving some of these challenges?
Yeah, I mean, it's hard to answer that question.
You get it a lot, I'm sure, but
yeah, in a general way, because it is a little bit disease specific. Certainly Alzheimer's disease has received the most attention from drug companies in terms of the neurodegenerative disorders. And I'd say one of the problems there has been that the companies maybe prematurely focused on one idea about how that disease is caused. And now they've done quite a few clinical trials to test that idea. And it looks like either the idea wasn't right or that there's no practical way to intervene in time to really, intervene based on that idea. So I think in that case, I think the field's really trying to regroup and think about other strategies to treat it. I think for me, one of the most exciting sort of developments within the neurodegenerative disease space is that for years, from the standpoint of therapeutic development, the focus has been developing a pill. We still want to do that, but I think there are a bunch of new exciting modalities for developing therapeutic interventions for neurodegenerative disease. And right now I'm thinking of something called antisense oligonucleotides. The details aren't important, but the idea is that instead of a drug or a small molecule, what you're doing is targeting a particular gene that may be responsible or contributing to the disorder. And there was a childhood form of ALS called spinal muscular atrophy, where this therapy was developed a couple of years ago. And the results have been just amazing. I mean, literally these children would die within a couple of years. And now with this new therapy, they're running around and alive. And I think for many years, drug companies were skeptical that that approach would work at all. And so the demonstration that it does, I think, opens up a lot of exciting new doors for a lot of diseases, both diseases that are clearly genetic, but I think this general approach could be used to target molecules in the brain that might be difficult otherwise to target with small molecules. So I'm excited about that. I'm excited about, still excited about stem cells. I think that we have really tried to harness them as tools to create human models of disease that might give us more reliable insights about how to find and treat some of these disorders. But I think there's great interest still in trying to use these cells as replacement parts. And we're working with a drug company around one of those ideas right now too. So I'm excited because I think that it's, you know, one of the problems right now has been the productivity of the pipeline for therapies has unfortunately been declining somewhat steadily over time. But I think some of these new treatment strategies open up new possibilities that hopefully will lead to good news in the near term.
Mm. And your sense is that as some of these discoveries become proved out, that biopharma and the rest will rush back in if they've left the space for now.
Well, I've already seen you know, drug companies really change their mind, even the largest pharmaceutical companies, around this this approach of targeting genes. And so many of them you know, made their empire based on small molecule drugs, but almost all of them now have either developed internally or bought other companies to be able to add some of these other treatment modalities. And some of these companies are really in the leadership. Even AI, you know, which is the sort of subject of this talk, just in the last 2 years, it's amazing how drug companies have really tried to develop these technologies internally and to also partner with folks like us to try to bring them to bear on some of their problems. So I do see, I think, you know, maybe it's because of the challenges and the failures, but I do see a greater openness in therapeutics companies to some of these technologies.
So let's talk about the Thinking Microscope. You had, is this an iteration on the robotic microscope, or maybe you could start there by talking about the robotic microscope and why?
Yeah, do you mind, Robert? I have a couple slides. Would it be okay to
Sure.
show? Okay. So I think this was your Newton.
That's Newton, yeah.
And some future talks,
Oh,
I
yeah,
guess.
yeah.
All right.
That would be my slide. There you go.
So yeah, so you know, thanks, Robert, for this opportunity, and thanks for the audience, for all of you joining. You know, if you were there for the first part, you'll know I'm a physician scientist, and I think one of the huge motivations for me in working on neurodegenerative diseases is meeting the families who've been affected by them. And I love Gladstone's tagline, "Science Overcoming Disease," because I think we do have a focus on translational science to a greater extent than many places. And over a decade ago, I was very fortunate. Tad Toby and the Correct Foundation came together to enable me to start a center in addition to my academic work that was really focused on translating discoveries in the lab to things that could make a difference in the clinic. And I think that forced me to really look carefully at what is the current state of the union and what are the problems and how can we solve them. And it really is amazing that it still takes about 13 years and over $1 billion to develop a new drug. And the other thing that's been discouraging has been that if you look at the productivity, the number of new drugs that get approved over time, it really has been in long-term decline. Many of the drugs, for example, that are used to treat psychiatric disorders are ones that were developed in the 50s and 60s, and they're still in use because we haven't found anything better. In fact, if anyone's familiar with Moore's law, which is a law in Silicon Valley to sort of to convey the exponential growth in the power of computers, someone has coined the term Eroom's law, which is the opposite for drug development, because instead of increasing every year, it's been actually declining. And so I think trying to think about why that might be, what contributes to it, and what role we can play in helping to improve it has been a motivation for me in parallel to the focus on these specific diseases and treatments. And in general, the pipeline for developing new drugs has largely depended on drug companies making drugs, but based on innovative discoveries from academia like Gladstone. Mm. And if you haven't read this book, Rigor Mortis, I recommend it. It's a book that talks about one of the reasons why productivity in drug development is less than what it could be. And one of the shocking observations that a number of groups have made is that as industry tries to replicate scientific discoveries described in academia, the replication rate has been shockingly low. In one paper, it was 10%, in another, it was 20 to 25%. So there is a huge number of discoveries that are reported that are difficult to replicate in the hands of other scientists. And if you know anything about science, the key to scientific progress is one group making a discovery that can then be replicated and reproduced by others so that the scientific knowledge base can continue to be built. So if we have trouble reproducing scientific discoveries, it's a huge problem because it means that people can't place as much faith in the discoveries that are made, and it takes a huge amount of resources. I
just want to echo your recommendation of the book. I know that you gave it to me when I first got to Gladstone, and it's really eye-opening. It's easy for a lay person to understand as well. So I think it lays out the dilemma.
Yeah. Yeah, so, and I would say nobody wants to tackle this problem seriously. NIH has certainly tried to force academic scientists to include new sections in their grants to try to write in narrative form how their design is going to be more rigorous and reproducible. But in fact, there's very little that NIH can do to sort of enforce this. And there's lots of reasons. The book goes into some detail about why the incentives aren't well aligned for supporting this effort. And so this is really my, a basis for my big idea for this Newton series. This is a problem that desperately needs to be solved. Nobody wants to solve it, but we really need to. And so the book talks about several sources for the problems. One is that experiments that most academics can afford to do are too small, so the results are often misleading. If you do look at a small enough group, just statistically, you can get fluke results that look significant, but would disappear if you did a larger group. You can think about it if you were flipping a coin. You might get 3 heads in a row, but obviously if you flipped a coin long enough, you'll get an equal number of heads and tails. And the problem in academia is that if people stop when they have a significant result and then publish it, then you only see these so-called significant results that really can't be replicated in a larger group. Another big issue is humans are humans. And so one of the things that humans do is they're not, everyone's a snowflake. So trying to get people to do reproducible science where they execute the experiment exactly the same way every single time is surprisingly hard. And so I think there's a huge role for automation in this process to help. And then I think another aspect of being human is just we're subject to various biases, and none of this is really malicious. It's just the nature of being human. We have observer biases or selection bias, publishing bias. And so if there were technological solutions that could help address some of these issues, that might be an approach to improving reproducibility in science. So Robert asked about this system we developed, robotic microscopy, showing you a movie now of a 4th generation system we built. Has a robotic incubator where plates sit with in the condominium. We've integrated this with robotic arms. You see a 4th generation system in the foreground and a 3rd generation system in the background. And then we built these fully automated microscopes and written computer programs that enable the system to be able to take pictures automatically around the clock, 24 hours a day, 7 days a week. And we programmed the instrument so that you can see those cells and follow them as individuals, like a patient, over time. It's been invented and developed at Gladstone, and we've used it extensively as a tool to understand neurodegenerative disease. And it really helps to address some of these issues that I raised, because it makes it easier for experiments to use larger numbers of cells so the results aren't so susceptible to fluke findings. It takes humans to some extent out of the experiment and out of the equation a little bit more, because you've got robots doing the work, and they can do things like boring observations and transfers much more reproducibly than humans. And because we take pictures of essentially every cell in the dish, and all of our analysis computer programs are automated, we can remove a lot of the bias that can affect scientists.
It's applicable across biology, not just for nerve cells, right?
Yeah, exactly. And I think that was what I was mentioning earlier. You know, we developed it because we were painfully aware of all the limitations in neuroscience. But once we developed the platform, drug companies in particular saw applications to other parts of the body, cancer and GI problems and other things. And so they have asked to partner with us to use this platform for those problems too, and it's very helpful. So that's something we invented at Gladstone years ago, and we've continued to develop it and add on capabilities. The big new idea for today's talk is really trying to bring artificial intelligence into the system in a greater way. So we've been working with Google for 5 or 6 years on using AI to analyze the data that those robots produce. But what I'm talking about today here is actually having AI power the microscope itself, even designing and doing experiments. And if you haven't been keeping up with this field, it's just astonishing, kind of the rate and progress and what it can do. There's one of my favorite stories is Google developing a program to play chess, and that's something that computer programs have done for a while. But what they did with AI was develop a system where the computer never was sort of taught the code or rules or moves like IBM, for example, did with its Watson program. Instead, it used AI to create a system that could just watch other people play and learn by watching. And what was so amazing was that when they then used this program to compete against, for example, IBM Watson, it was much more successful, much faster. But the most exciting thing was that it actually made some moves. If you follow the chess world, there's game 7 of this particular matchup, where the Google chess program did a move that humans have never seen before. And what I think is so cool about that is it suggests that AI, that it understood kind of a truth about chess that even humans couldn't grasp. And we've had this experience before as well. We've published deep learning AI approaches to analyzing images, and it can do better than humans in many cases, do things humans can't do. So there is the possibility of discovering.
It suggests a different term, maybe this is out there, but of artificial imagination.
Yeah. I mean, it's, where exactly creativity and imagination, how to define that, I think is a really interesting question. And I think you're right. So that, in fact, is one of the human biases that I'm trying to solve here. So Google actually thinks that the main limitation or bottleneck to science is the human brain and the ability to conceive the questions to ask. And so if you, with AI, the wonderful thing is it doesn't have, it's not burdened by those biases or constraints. It's not burdened by the knowledge base that it has already, or even hypotheses that make sense to it or hypotheses that appeal to it. It has the possibility of being able to explore biology in a way that lacks those biases. And so what we're trying to do is build a thinking microscope where AI would actually control the microscope and do the experiments. And what we think this will enable us to do is to be able to use light to be able to stimulate and induce experiments in individual cells in a well. So massively parallel, we'll be able to do many more experiments at once, even in a single well. And we'll be able to have the machine learn from watching what it did to design the next experiment even before we finish that whole experiment. So we think this will rapidly accelerate the whole process and make science faster, cheaper, and with a lot less bias.
What are we seeing at the bottom of the screen with the graffiti?
Yeah, so this is an example of what we're proposing to use to help. So with experiments, you need some way to perturb the system to basically trigger the experiment. So you're trying to do some sort of stimulation, and then the experiment is to observe the response to that stimulation. What you're seeing here is a hardware device that's capable of rapidly directing a beam of light. And you're looking at in this case, just even a single cell that's been labeled with a fluorescent label. And the light is drawing the name Mike by bleaching that fluorescent molecules. And it's just meant to show that we have the technology to be able to deliver light to precise locations extremely rapidly. And there now is a whole set of tools available for triggering biological perturbations just using light. So people have figured out, we and others, ways to use the light photons to be able to trigger biology. So on the right here, you see this circle, which is kind of the cycle for deep learning. The idea would be to collect an image. The computer using AI would look at the cells and make some decision about how to stimulate them. It then would design the stimulation with light, and that light would trigger biology that we would then follow over time and repeat the process. So in the course of maybe a week or two, we could do millions of experiments on individual cells, explore the biological network in a much greater depth and breadth, and design experiments on a per cell basis to be able to accomplish all that. And so we think this would completely change the way science is done, not only in terms of making it faster and cheaper, but also I think being able to discover biology in a much less biased and broad way that hopefully, as I said, will lead to a better understanding of the causes of disease and ultimately treatments.
Can you say something about the importance of longitudinal data and maybe a specific use case where this platform has helped us understand how cells degenerate and lead to Alzheimer's, or how is it that we're leveraging this narrative of the cell's life versus a snapshot?
Yeah, well, I'm sure the audience can just in their imagination think about how much more information there is in a movie about someone than there is in just a single photograph of that individual. And so I think what we've done by building the initial robotic microscope, even before what we're proposing to do here in integrating AI, what we've done is create a way to be able to generate many, many movies of individual cells. And what that's enabled us to do is to watch disease as it unfolds in a cell and to better understand how things that happen to a cell early eventually determine what happens to that cell in terms of neurodegeneration. And maybe the one example is the very first application of this instrument. We were trying to address a question that had been in the field for a decade, but was unresolved because it was impossible with snapshots to address it. And that had to do with the role that protein deposits play in neurodegenerative disease. And the surprise was that when we made the movie, So before, just based on snapshots, people thought those deposits were the cause of disease. When we finally made the movie and saw what the relationship was, it was just the opposite. It turned out that those deposits seemed to be an effort by cells to sequester those disease-causing proteins into a form that was less troublesome to them. So I think it highlights just the potential here for sometimes coming up with results that are completely the opposite of what you would get with conventional approaches, and then finally being able to have a basis for making more reliable decisions about how to intervene.
What's the barrier to building this platform? Is it just manpower, resources? Is it just you're still trying to solve problems as you go, or what's the?
Yeah, so first of all, I just want to thank the Correct Foundation. They have given us some seed funding to help to advance efforts in this area. So I think we've begun to start the process, but certainly part of it is resources. Some of these technologies are expensive from a hardware standpoint to bring together. Another aspect is simply it's never been done before, and so we do have to develop new algorithms, computer algorithms, to be able to do this. And as with any engineering project, sometimes the obstacles get discovered along the way. So as you try to build these things, you realize or uncover bottlenecks. And I think one particularly interesting challenge is the rapid communication that has to happen between the AI network and the microscope that it's trying to control.
It is a pure bottleneck that it can, yeah. We got about 15 minutes left, and I made the mistake last time of not accounting for the questions that would come in. And Sarah's going to help us go through some of them that come in as we've been talking. Sarah, you want to just kind of screen through the questions and
Yeah, no, we got some really good questions so far. Thank you guys. And if you have any more, please type them in the chat box right now. But the first question is, when the science research offers no solution, how does your faith inform you, if at all?
If we're talking about a scientific problem, I think my faith gives me the courage to keep trying. I think one of the big challenges in science is that even on a good day, a lot of what we try doesn't work, in the sense that we don't get the answer we want or a clear answer to the question. And so I think that I think for me, all the contact I've had with families and my faith help, I think, give me the motivation to hang in there, because I think a lot of people have found that the number one kind of personality quality that predicts your success in science is resilience. And so I think being resilient is important.
You know, I heard you at a talk a year ago talk about the number of the consecutive failures. And something you said really struck me is, you said, we know where the mountain is, just don't know how to get up it, but science is the way up, right? So
yeah, no, that's a good point, Robert. I think, I sometimes I think of Mount Everest because people think about that as sort of the hardest thing they can imagine, but no one has cured any of these diseases. So we know there's a problem, but we don't even have the path well defined, and we don't have a sense of how difficult it ultimately will be. But there really is no path up the mountain except through science for solving these, at least developing therapies.
Great. Great. So the next question kind of relates to the discussion earlier, but what steps and precautions do developers at Gladstone take to make sure that the human bias is not directly programmed into the AI microscope?
Yeah, that's a great question.
Really
And probably question. Some folks are aware that, people have looked, for example, at AI and applications it's had to hiring people and, other sorts of applications, and have discovered that that some of the ways that people have developed these algorithms unwittingly introduce bias into the algorithm that ultimately gets produced. So it's a really interesting question. I think let me say two things. One is that one advantage to being able to write automated programs to do the experiments and the analysis, especially on the analysis side, is that it could be that we have some bias or at least some error that has been introduced into those algorithms. And I think one of our goals, in terms of reproducibility, is being able to write those programs, but then give them away to the scientific community. So we have repositories where we can put these algorithms and let other people use them. And what we want is we want people to apply them, to see the results they get with them, and to decide for themselves whether they think those results are good quality or whether they think that there may be an error in there. And I think that sort of feedback would be very useful for helping us discover whether we've inadvertently introduced some error into the computer programming, and hopefully over time be able to continue to refine and improve them in a way that will ultimately get the best tool we can. And at the same time, make all of the things that we do very explicit, because that's one of the big challenges with human bias, is that people make up their minds about sort of what their judgment or decision is, but exactly how they made their decision isn't explicit. And I think the one advantage with a computer program is that we can do that.
Great. So the next question is, can you continue to do lab work during the pandemic?
Yes, we do some. So we have been designated as essential work. And so we have the authorization from the government to be able to continue to do work in the lab. At the same time, obviously the safety of the folks who work in our group is paramount. And so we want to do this in a way that keeps people safe and poses the minimum risk. So we've tried very hard within our group to identify the key things that need to be done, but to limit our work as much as we can to just those essentials. One nice advantage, though, I will say, is that because we do so much computational work, that is something people can do at home. And so we have had many Zoom meetings. We just had one yesterday. Our AI group was talking about new algorithms and exciting new results. And unfortunately, we can do that at home as individuals. And so we can continue to work in some areas almost unabated.
And this next question is actually kind of related. So in the start, you mentioned about outreach and how much you enjoy it. Would you ever consider making an online course on how to build algorithms to analyze images so that maybe more labs could be accessing these images if they don't have availability to microscopy or in their lab?
Yeah, it's always a challenge in science because the people who pay our salaries pay us to do research. And so it's been difficult to get the time to be able to set aside to do kind of what you described. But we have had folks in our lab who have taught a graduate course for UCSF in an effort to try to make some of these approaches available more broadly. Another approach that we've done has been to create online repositories with some of these analysis tools, as well as essentially some tutorials there to help people. So I'm really sensitive to that. It's a great question. We desperately would love to disseminate this. Sometimes people just decide that the best way is to join our lab for a while and learn, from doing it.
Great. So another question we got is, how do you estimate how many iterations are required, assuming we're talking about ML-driven optogenetics to produce a result? Is this a combination of search space and statistical significance?
All right, I've got an aficionado in the audience.
That was my question, actually.
Yeah. So for those of you who aren't AI folks, what they're asking has to do with, so one of the things with deep learning, machine learning, is that, especially the convolutional neural networks that we use for a lot of our work, is that they're powered by massive amounts of data. So a lot of people think about AI as a tool to analyze massive amounts of data, but in fact, a lot of these tools absolutely require it to even learn. And I think one of the reasons Google, we were apparently their first academic collaborator, and the reason they said that they came to us was because we generated enough data to be interesting, was kind of the way they put it. So I think the question is, how much data do you actually need to get something useful? And it's hard to answer that in a sort of general way. And it's not something we've looked carefully at in too many specific cases, but I can give you one example. There's a paper we have on bioRxiv where one of the postdocs, Jeremy Lindsley, who had developed a new biosensor to tell us the very first time point when a neuron is committed to undergo neurodegeneration. That's really useful information for us as we study neurodegenerative disease. And it turns out humans are really lousy at it. We've done this experiment lots of times where we've had humans try to curate cells, and they don't get it right very often. So he developed this biosensor to be able to identify that time point, but then he wanted to see if he could train a deep learning AI network to do it just using the shape of the cell. It's sort of the information humans tried to use but weren't very successful. And it turned out that he could develop a deep learning network that was essentially 99% correct based on morphology. So evidently the information was there. It's just humans don't seem to know what to look for to be able to make that judgment accurately. And when he looked at how much data he needed, it was about 15,000 images before he saw some really substantial learning or improvement in performance. So these approaches do require large amounts of data to be able to develop the algorithms, but the good news is that once those algorithms are developed, they can be applied to smaller data sets. We've also shown that you can do something called transfer learning, which is to say, if you do train a network to do one task, sometimes you can train it to do other tasks that are somewhat related, new tasks, but somewhat related with much smaller data sets. And there's a great amount of interest in the field for being able to figure out how to train on smaller and smaller data sets, because particularly as it relates to clinical disorders, we often, when we're following patients, we don't have 23,000 patients. And so we have to find ways to be able to do this better.
I'm sure there will be other questions. Steve, if you can just flip to my end slide where we give my contact information. As people have follow-up questions, I can collate those and get them to Steve, and we'll make sure we respond. I thought we might end, when we interviewed Benoit, we asked him 3 questions just as a speed round, and we thought we might continue that tradition. And I actually can't remember the questions we asked him, but I wonder, what would you be if you weren't a scientist and a doctor?
If you hadn't included scientist, it probably would be a physicist. I have 3 boys. 2 have gone to AI grad school, and the 3rd is an astrophysicist at Berkeley. And so I think there's something in the genes. But if you took all that off the table,
yep.
You know, there's a part of me that there's a creative side to me that I just love science because it does offer me an outlet to express that. In a way, to be honest, it's hard to express in medicine. So I do like that. And so that creativity gets expressed in other places. I love to do gourmet cooking. I do a lot of woodworking. I build stuff. I've been painting lately. So maybe something in that neighborhood.
What do you think, where do you think medicine will be in 25 years?
Well, I think that certainly over the next 10 years, we and others have gotten better at leveraging all the genomic data from sequencing the human genome. And I think as we do a better job of using some of the tools even we talked about today to leveraging electronic medical records and other large data sets, I think we're going to get a much better understanding of how the genetic seeds we have in our genome predispose us to the risk of certain diseases. And I think that's going to be just a, I think the whole area of functional genomics, where we're trying to really understand the functional implications of the genetic variation in our genes, is going to be a treasure trove when it comes to understanding our personal risks for disease, as well as the causes of disease and developing therapies. And so I think it's probably, not a shocker that I think personalized medicine will, become much more mature. And I think, one thing that has been almost an impossible nut to crack has been understanding environmental influences. So we know, because we can do the kind of genetic experiments to understand how heritable certain conditions are. And we know that there's a portion of disease that's, at least we can't detect heritability. So we've assumed that there's some environmental influence that's present in those disorders, but it's been very tough to study. Part of it could be just bad luck. But I think that as we have a better, more specific understanding of the genetic underpinnings of disease, it's also going to make it much easier to understand environment. And there have been some really cool studies too, where, in Iceland, for example, where the population is relatively small and they literally have studied every single individual, where you can even show that the chromosome you didn't get from your parents actually can contribute to the environment that they create to educate you and raise you and influence you. So it's even possible some of our environmental influences come through genes, but genes we don't get directly.
Oh gosh, that's fascinating. Well, we're up against time, and we want to get you back thinking about your science. The last question is just about culture. One of the things I know we all miss is just bumping into each other in the lobby or walking to the garage and asking, what are you paying attention to these days? What is it over the last couple of months? Is there something that you've encountered, you've read, or you've heard, or you've tasted that is memorable and you'd highlight?
Yeah, what an interesting question. Well, I finally, one nice thing about shelter in place is that I finally had enough time to do a decent sourdough starter. That takes a lot of effort, and it's like a pet. It's almost like a baby, if anyone knows how to cook these things, it's crazy. And so this has been a lifelong ambition for me, being in San Francisco, and finally I've accomplished it. I think it could be improved, but I definitely am. Something I've tasted that I've really liked.
I am so surprised that you're not the only person. I must know 15 people that have been geeking out on their sourdough starter. Maybe it's just where we are. Well, Steve, I miss being able to see you and hang in there. And thanks for taking time for this.
Yeah.
And any questions we get from this, we'll forward along to you.
Great, thanks. And thanks to everyone who showed up. Really appreciate it.
Thank you all. Next week, it's Jean Paul's on epilepsy.